Friends, welcome to Biology Made Easy. Today we will discuss the second part of this cell cycle. In part 1, we have discussed what are the causes of cell division, why cell division is required for the growth, reproduction and repair. All those things are clear. Then toward the end of that video, we have discussed that the cell cycle is divided into interphase and M phase. The interphase is very long, and again it is divided into three substages, that is G1, S and G2. Interphase divided into G1, S and G2 phase. Now what are the different events, sequences that take place during this phase that we have to discuss. But here I want to clear one thing, because most of the students often they ask doubts. Suppose the cell is not dividing, then what is present? What is the structure of the chromosomes? And when it is dividing, what is happening? That we have to discuss. Suppose let us take one example, onion. In onion, 2n is equal to 16. That means this is the cell, this is the nucleus, here 2n is equal to 16 is there. That means 16 number of chromosomes are there when the cell is not dividing. Then how many chromatids are there? The chromatids are also 16 number of chromatids. And how many DNA molecules are there? Again 16 DNA molecules. What is C value? If this since this is a 2n diploid cell, so the C value is again 2C. Now, when the cell will be doubled, everything will double. The chromatid will be doubled, the DNA will be doubled, C value will be doubled, but not the number of chromosomes. This is one example. Then we can take the example, another example from human being. Now you take the case of human being. In case of human being, 2n is equal to 46. That means 46 chromosomes are there. That means how many chromatids are there? 46. How many DNA molecules are there? 46. 46 DNA molecules are there. And what is the C value? Since 2n, we can write it 2C. But this 2C and this 2C are different because here the haploid set is only 12. So its weight will be different. Here haploid is n, so its C value will be different. I have written 2C because 2n. That doesn't mean that the 2n is same, the 2C is same. It depends, this 2n is a number, while C is C value, it is the weight of the DNA. So this is known as C value. And in each chromosome there is a single DNA molecule. When the cell will undergo these different stages, the chromosomes will condense, and each chromosome will replicate during S phase, then only it will become double. So suppose you take the human being, so here 46 chromosomes are there, now it will be 46 into 2 chromatids will be there, 92. And here 46 DNA molecules there, then it will be 2, there will be 92 DNA molecules. So from this point it will be 92, and it will go on, again it will come to 46 at anaphase, then again that 46 will continue. This is the main reason. That means the number of chromosomes always remain constant, 46 throughout 46 it will remain. But the number of chromatids, the number of DNA molecules may be doubled, and the C value may be doubled. Again, at some place like anaphase, it may be reduced to half. But the chromosome number always remains constant unless it is meiosis. This is the basic idea to understand, so that you will be able to answer almost all questions in cell division. Now, let us come to this first stage, that is the G1. From this cycle you can mark that is G1 is the biggest stage. It is the, you can tell this is the longest stage. So what are the different characteristics of this G1 phase? We discuss one by one. My first is, this is known as the first gap period. This is first gap period. That means this period is between the M phase and the S phase. So it is known as first gap period. is also known as post-mitotic stage. Post-mitotic, that means after mitosis complete, this stage is occurring. Here again some students confuse just at the beginning why we call it post, why it is not pre, because this is the M phase. Any division, anything that is taking place before it will be pre, but after it post. That is why this G1 is also known as post-mitotic stage. This is the longest phase of the cell cycle as well as the interphase, because it covers about 40 to 50% of the cell cycle time. Suppose you take the case of human beings. In human beings, the entire cell cycle is 24 hours, of which 23 hours only interphase. And out of these 23 hours, 10 to 12 hours only this G1 phase. That is the longest phase of the entire cell cycle. Now, the next thing is what happens during this G1 phase. So this is one cell. Now it will divide into. So initially the size of the cells are very smaller. So naturally they will grow in size so that they will retain their original size. That means cell growth takes place so that the size becomes double. The size becomes double. The original size will return back. Then what is the next point? The chromosomes are long, thin, and less coiled. Again here the students confuse when the chromosomes are less coiled and when more coiled. This is a confusion with students. Now here, because students they think that during this interphase the chromosomes are not clear, that is why they are more coiled, but actually not. The chromosomes are actually like this. The chromosomes or chromatin, whatever you can tell, they are like this. They are less coiled. They will be more coiled only during metaphase. That is why that will be visible. So this is also very basic idea. The chromosomes are very long, they are thin and less coiled. Then the next point. Now, what are the different events that take place during this stage? It is very easy to remember from examination point of view. Why? If you remember one thing, what happens next, then automatically everything will come to your mind. In S phase, the replication of the DNA takes place, and you know for replication what are the different requirements. So if you write all those things, your work is over. Since replication will take place in the next stage, that means the cell has to prepare itself in G1. That means it synthesis of nucleotides. Unless nucleotides are synthesized, how polymerization will take place? So that is number one. Again, you know that for replication, several enzymes, helicase or unwindage, topoisomerase, then DNA polymerase, primase, so many enzymes are required. So synthesis of enzymes. Next, synthesis of proteins. But remember, these proteins are always non-histone proteins, not histone proteins. These proteins are always non-histones. Non-histone proteins are synthesized. Then, when proteins and amino acids, enzymes are synthesized, unless there is formation of amino acids, how that will take place? So, next is synthesis of amino acids. That will take place. But how the synthesis of enzymes, proteins, amino acids will take place? Unless the DNA transcribes RNA. So that is again important point. You can write synthesis of RNA molecules. Synthesis of RNA molecules. So now you see, that means whatever is required for the next stage S phase, all these are synthesized. The nucleotides, the enzymes, the proteins, the amino acids, the RNA, everything are synthesized, but still replication cannot take place because replication requires a lot of energy in the form of ATP. That is why the next step is synthesis of ATP. Because without this energy, replication cannot take place. Further, during M phase, in anaphase also it requires a lot of energy for separation. For separation of each chromosome, it requires 30 ATP molecules. The more the number of chromosomes, the more the number of ATP molecules required. So the ATP molecules are also synthesized during this G1 phase. Now you see, when synthesis of so many things, amino acids, nucleotides, proteins, enzymes, ATP, RNA, all these things are taking place, how can you tell that the G1 phase or the interphase is inactive? When so many things are produced inside the cell, how can you tell that it is inactive? That is again next question. During which stage of the cell cycle it is very, very active, or during which stage the gene action is more? The answer is G1. That means maximum gene action takes place during this phase. That is a very, very important question. So now everything is ready. But whether the cell will divide or not divide, it has to take a decision. If it has to divide, then it will synthesize all those things and it will go to the next stage. But suppose you see, this is one cell. Now this divides into two cells. Is it compulsory for them that again both cells will divide? No. Both cells may divide, both cells may not divide, or one of them may divide, the other may not divide. You may ask how? You see, suppose this is a plant and at the tip of this there is a meristematic tissue. If all the cells it produces will be meristematic, then the entire plant will be meristematic. How will get the permanent tissues, parenchyma, collenchyma, sclerenchyma, how will get all these things? That means when cell divides, suppose divide two cells. Suppose this cell will divide into two cells, this may not divide. Again, out of the two cells, one may divide, the other may not divide, so that some cells become permanent, while some cells remain meristematic. They will again divide. That means the decision of this cell, whether it will divide or not divide, that taken at this point. Decision of cell division takes place during this G1. Now, suppose this cell will decide not to divide. It wants to become permanent. Then it has to exit the cell cycle. This is again a frequent last question. Now the cell has come to this G1 stage. Now it decides not to divide, it wants to become permanent. Then it will exit the cell cycle. It will go outside. And when it will go outside, this is known as G0 phase, or you can call it as Quiescent phase. That means this cell, when it decides not to divide, it exits the cell cycle towards the end of G1 phase. It exits and it enters into a new phase which is known as G0 phase or Quiescent phase. So at that G0 phase, these cells are metabolically very active, because unless they are metabolically active, how they will survive, how they will live, how they will perform their activities. Metabolically they are active, but they do not proliferate, they do not divide, unless called on to do so. Now listen very carefully. Now suppose the cell decides not to divide, so it will enter to G0 phase. You can take the case of parenchyma in case of plants. It become differentiated. Then when it moves to G1 phase, it undergoes a differentiation. Differentiation, that means it becomes permanent. It will not divide. Differentiation. But if required, if called on to do so, then it will again return back. It can return back again to this G1 phase if it is required. Suppose in our body some injury is there, then how it will be healed up? Only the cells divide. That means it was in G0 stage, but it has to return back to G1 phase. Again it will prepare itself, then it will undergo division, so that our injury will be repaired. So when it was in G0 phase, it was differentiated. Now again it will return back. We'll use the word de-differentiation. These terms are also very important in anatomy. Differentiation and de-differentiation. Now I am giving one example. Suppose let us take one dicot-stem. In a dicot-stem, these are the vascular bundles, and in vascular bundles there is in between xylem and phloem there is the intrafascicular cambium, like this. But when secondary growth will take place, it gives rise to a ring of a cambium. You see, in between two vascular bundles, this is medullary rays, which is made up of parenchyma and osteocytes. They are permanent, they are in the G0 phase. But when the cells will divide, these parenchyma and cells lying in between the two vascular bundles, they will also become active and divide, like this. They will be active and divide, and a ring will be formed. So this is known as interfascicular cambium. Now how this interfascicular cambium is formed? From the permanent cells by the process of de-differentiation. When they were formed by the process of differentiation, now they return back to meristematic condition. So by the process of de-differentiation. And now this interfascicular cambium, it will divide to give rise to secondary phloem toward the outer side and secondary xylem toward the inner side. So previously primary xylem, primary phloem were there, but now the new tissues will form towards the inner side, that is secondary xylem, toward the outer side, that is secondary phloem. That means when differentiation to secondary xylem and secondary phloem takes place, that is known as redifferentiation. Redifferentiation. So from G1 when it enters G0 phase, differentiation. If it is called on to return back, like at the time of injuries, then it is de-differentiation. Again, when it will give rise to new cells, it is redifferentiation. These three terms are important at this place, again they are important in case of anatomy. So, if the cell decides not to divide, it will enter into G0 phase, and all these things will take place. But if it decides to divide, then what will happen? If it decides to divide, that means from G1 stage, it will go to S phase, but not so easily. Here, before entering to that point, here it will face one checkpoint, which is known as restriction point, or you can call point of no return, or you can call rate limiting step. That means before it enters to S phase from G1, everything is to be checked. There are different checkpoints. We'll discuss it separately because that is very, very important. But now let us remember what are the different events that are taking place during this is G1, and all these things I have written in a series of points. And I hope what are the different things that are taking place, what is very active, what are produced. You can remember very easily once you know that the DNA is replicating in this space. All these things very easily you can remember. And these are very important. The G0 phase, point of restriction, what are the different points, what are different things that are produced. All these are very, very important from examination point of view. Now, what is happening in the next two stages that we'll discuss in the next video, but here I will give you questions. So one question you can write. One question. At which stage maximum gene action takes place? So A choice G1, B choice S, C choice G2, D choice M phase. Number 2, synthesis of proteins and RNAs takes place during A point G1, B, S, C choice G2 and D choice M phase. So I hope you have enjoyed the video, and again I hope your concept is now very clear. You will not commit any mistake in cell division. Still more points are there ahead in other steps. So I hope you have understood everything perfectly. Thank you very much for watching the video. Have a good day.